Can everyone up here see on this screen the vagina, the oviducts, and the, well, a couple of ovaries and the fallopian tubes are oviducts? Everyone okay with seeing that? No? We need to turn the lights down a little better? Do you mind, Rebecca, reach through Harry's pelvis over there and hit the bottom switch?
Okay, I see.
You don't see the vagina?
Nope.
Well, just a minute ago you were saying yes. No? Just checking. Okay, so none of you will be the guilty parties next week.
Oh no, that is horrible.
And we'll mix up male and female reproductive systems. It happens every year.
Really?
So I just want to make sure, right, that no one's seeing what I just said, right? This is in fact male, and labeled on here would be a penis, and the vas deferens or the ductus deferens, and a couple of the testes. Okay, let me just walk you through the major structures for this. Does everyone in front of them have a male model? Okay, everyone should have a male. If not, grab one. They're right over here on this table. Sarah, there's one for you. And then there's another male model. There's a hanging system. There's two of those in the room. I see those floating around. But every table right now should have a male and a female. And we'll get, right now all you need is a male. So what's missing here, of course, would be the scrotum. And that external tissue has been removed. So we're looking at, in this cartoon at each testis. Now, I want to make sure that we're okay with the spelling here. This is a testis. T-E-S-T-I-S, right? Testis, one. If we're talking about both of them, it's the testes. Okay? E-S. I don't know what this is. Okay? That's just, you're just a little confused. Okay? So none of these, right? One testis or two testes. And when you cut into the testis, what you would see is a very remarkable series of tubing. Those tubes are the seminiferous tubules. There's yards and yards of tubing within each testis, and it's in those tubes that sperm are being produced once puberty comes along. Now, once the sperm are first produced, and I'll show this to you in a moment, they're not swimming yet. They just kind of get washed away by the current of the fluid, and they're going to go to a storage vessel called the epididymis. Epididymis is sitting on sort of the superior on the side of the testis. Now, the sperm will be stored there for a short while. They'll kind of grow up a little bit, mature a little bit, learn how to swim. It's swimming school. And then either will be resorbed by the body, recycled, or upon ejaculation will be released. And the sperm will go up the vas deferens, basically come back into the body cavity, right? So they're coming from the scrotum out of the body, back into the body proper, come along the bladder, and join up with a gland called the seminal vesicle. There's two of them, one on each side. Now the seminal vesicle is going to be producing the majority, not all, but about 60 or so % of the fluid that's found in the semen. Now the vas deferens, also called the ductus deferens, this tube bringing the sperm up, merges with the seminal vesicle and drops both of those, and drops now that fluid and the sperm into the urethra through a, we'll see it in the female as well. This is basically, we know a diaphragm is something that separates the inside, or separates, right? So the diaphragm of respiration separates the thoracic cavity from the abdominal pelvic cavity. This diaphragm, the urogenital diaphragm, separates basically the outside world from the inside world at the urogenital region. So it's a band of muscle, and let's think about it. What muscle would we find that we talked about last week here at the base of the bladder entering into the prostate? Remember, there are two sphincters, internal and external. So there at the base of the bladder going into the prostate, that would be the internal urethral sphincter. That's smooth muscle. It's involuntary, giving one the urge to go. Then in this same muscle layer of the urogenital diaphragm would be the external urethral sphincter, giving one control over urination. That one is voluntary skeletal muscle. Okay, so those are the major structures, and we'll take this information and, as we have been, and look at the model in a few minutes. I want to give you a little bit more detail here just to pull this whole story together, looking at some of the questions that you posed. Perhaps this will help with some of those. The three glands that are producing the semen and the seminal fluids are as follows. So let's just go through these next 3 slides. Now, I would not be asking you, as you become accustomed, direct questions about this on your quiz next week. However, you know that reproductive system is going to be one of the 3 lectures on the final exam. And that's going to be, tomorrow I'll be discussing the respiratory system with you in lecture. Then next week we'll have 1 lecture on the urinary system, and then finally the last lecture on the reproductive systems, male and female. So I'm telling you some of this again to get our mind thinking about this. That's all we have left, right? Your cardiovascular system exam is behind you, and really all you have left is a lab exam and this final exam covering these last 3 systems. So the seminal vesicles, you see them, they're the little mustache on the base of the bladder in the urinary model. And there's 2 of them, left and right. They're coming up alongside the bladder. And the vas deferens or the ductus deferens is going to merge with the seminal vesicle and drain that sperm and seminal fluid through the ejaculatory duct, and into the urethra, into the prostatic urethra. This seminal vesicle fluid is viscous, it's thick, it's a whitish yellow in color, it is alkaline in its pH, and it contains fructose, fruit sugar, and it contains prostaglandins. Now each of those components is important for overall reproductive quality. So the viscosity of the semen is important because upon ejaculation the sperm must basically stick to the vaginal wall, and we'll talk about that in a moment. The alkalinity is important. The vagina is quite acidic. That acidity is important to maintain for normal antibacterial and antifungal infections. And so if you took sperm and put them directly into the vagina, they would just go belly up. They can't handle that acidity. So the semen must be alkaline to help neutralize that low pH. Okay, so we've talked about alkalinity and neutralizing fluids back in lab 2. The fructose is basically the granola bar for these sperm. They have a long trip ahead of them. They're very, very small cells, as we'll see today under the microscope. They have a very long journey ahead of them based upon their size, and they have to get up through the vagina, through the cervix, into the uterus, along the fallopian tubes, and all the way to the ovary to fertilize. And so they need some sugar and some energy, and that's what the fructose is there for. Finally, there are prostaglandins. Prostaglandins are smooth, muscle-relaxing molecules. And the cervix, the neck of the uterus, is usually closed down. And the sperm must penetrate through that door, and so the prostaglandins are going to help widen and dilate the cervix for the passage. So each of those components is important for overall functioning to go well. Now there's also the prostate gland. There's just one of them. We've seen it. It's that walnut-shaped gland. The urethra passes right through the center of it. It's on the inferior border of the bladder. It's also secreting fluid, and it is stronger. It has, citric acid, another energy molecule. It has seminal plasmin. This is a naturally occurring molecule which basically acts as an antibiotic. It helps to reduce the number of UTIs in males. Women have 2 disadvantages, if you will, as far as UTIs. One, the urethra is very short. The male urethra is longer. So men are going to have fewer UTIs for that reason. And the seminal plasmin is helping to reduce infections as well in the men. Finally, there's a molecule called PSA. PSA is something that is screened for often in a routine blood workup for a male, especially after the age of 30, or if he has a history of prostate cancer in his family. And PSA is a naturally occurring molecule in the prostate, but it's used as a marker for an enlarged prostate or a cancerous prostate. So the blood levels are taken of PSA, and if they're high, okay, now we're worried, is the prostate enlarging, which is not uncommon after the age of 50, or is it something worse? Is there cancer going on here? In which case we do more tests. Naturally, the PSA is an enzyme, and it's going to help liquefy the semen following ejaculation. So let's put all these factors together. So ejaculation, the alkalinity helps to neutralize the pH of the vaginal secretions. And then the viscosity is going to help the semen stick to the vaginal wall. Those prostaglandins are going to start coaxing open, opening up, widening up the cervix, and then shortly thereafter the PSA will come along and liquefy that viscosity and allow the sperm now to move out of that semen and move toward their goal. The sperm are directed both by heat and by chemicals. So they have little heat-seeking and chemical-seeking missile, basically, and they're moving toward the warmer temperature of the uterus, and they're also being drawn there by chemicals. The third glands are the bulbourethral glands. Again, first described by a guy named Cowper, so he gets his name thrown on them. They are on each side of the spongy urethra, right along that urogenital diaphragm, and they are providing a clear, mucousy-filled secretion, basically pre-ejaculatory fluids. And there's two reasons for these, at least two reasons for this pre-ejaculatory fluid. One is to cleanse the urethra of urine, right? Urine is typically acidic. And I've already told you that sperm don't like low pH. So in order to cleanse the urethra of urine, the pre-ejaculatory fluids are going to first cleanse that and then also act as a lubricant for intercourse. So you've got at least two purposes of the Cowper gland secretions. So those are the three glands, five glands altogether, right? 2 seminal vesicles, 1 prostate, 2 Cowper glands that are contributing to the seminal fluids in semen. Any questions on those glands or anything in the male reproductive structurally?
Is there a reason why the prostate gland grows after the age of 50?
I don't know that we know why. I'll show you a picture in a moment, and it'll help make more sense of it. But as we discussed last week, if the prostate does enlarge, right? I mean, it's important. It's important that it releases products of semen. And so that's its purpose, right, is to release those products. Its location, however, makes it difficult. So again, if that prostate's enlarging, which is not uncommon after the age of 50, then it can start pushing up on that internal sphincter, giving the urge to go when you don't have to go. It can be squeezing and making flow inconsistent, and it can even push down on the external sphincter, giving problems with control of urination. And finally, it can squeeze and interfere with ejaculation and with the ejaculatory duct. And so those are all the concerns, right, that can happen with an enlarged prostate, or if a prostate has to be removed, okay, because of cancerous changes. But why we have it, I guess you could argue, why do we have an appendix or a gallbladder too? They are things that we need, but we could do perhaps without, right? They're not perfect necessary glands. Let's take a look at, this is a cartoon of a blow up into the seminiferous tubules. Remember, it's in the seminiferous tubules where the sperm are being produced. And we've got 4 slides to look at today. One of them on the far table is going to be a cross-section or a section through the testis. And what you're going to see basically is a very crowded environment. You're going to see tube, tube, tube. Lots and lots of tubes. And in those tubes, there are going to be cells around the outside edge. Okay, you're going to see these cells. They're going to stain darker on the slides. So as best as I can draw this, little cells around the outside edge. And then in the lumen, in the opening of the tube, you're going to see sperm. And they're all entering into the lumen. It looks a lot like this. Okay. And that's what you're seeing here. This is the outside of the tube. So imagine a huge tube. And these are the cells around the outside I'm going to tell you more about in a second. And then here you have your sperm entering into the lumen of the tube. Okay. It'll be much more clear, I think, when you look at the actual histology. Now, this is the whole process here is the process of spermatogenesis. What we're looking at is the process of making sperm. This should make sense. Sperm, genesis in the beginning, the formation of. So we've got spermatogenesis, the process of making sperm. And what happens is that there are cells in the outer edge of the tubes. They've been sitting there since before birth, waiting for puberty to start. And these cells are spermatogonia. Spermatogonium. Each one's a spermatogonium, and many of them spermatogonia. Millions of these cells around the outside edge of every seminiferous tubule. Now, these cells are waiting for puberty. And at puberty, they start to divide. Now, notice that it says 46. That means that this cell is a diploid cell. It's like any other cell in the body, but its job is going to make sperm at puberty. So it will divide, and it will be dividing by normal ode mitosis, right? Just making another exact copy like itself. One of those daughter cells will be there to replace itself. The other one will know to continue and go through the process of meiosis. And remember, meiosis is a special type of cell division that is only used in the production of sex cells, egg or sperm. And the end goal of meiosis is to create haploid cells. Notice all these say 23. That means they have one copy of all the chromosomes. They are haploid. And in the process of meiosis, the chromosomes, remember, every cell in the body, you have some of your chromosomes from mom and some of your chromosomes from dad. You got one copy of each gene from mom and dad. And in the production of the sperm, you're mixing and matching that collection. And so each sperm is genetically different and different combination of genes from mom or dad and are haploid. The one cell that starts this process divides twice, and as a result, you get 4 genetically different sperm cells. Now, there's a really cool thing that happens here, and that the cells go from a little spherical cell to what we recognize as a sperm cell at the bottom, and I'm going to describe that for you very quickly here in a moment. But the overall process is spermatogenesis. Now, where does the testosterone come from? Right, we know the testes produce testosterone. Well, it turns out that there are cells, and in the cartoon they're on the top, interstitial cells. And we've seen that word interstitial before. Interstitial fluid was lymph fluid. It was the fluid between your cells. Here, the little interstitial cells are drawn in these little areas between the tubules. Okay, so outside of the tubules, but in between them, right? Interstitial cells. Now, these interstitial cells were described by a German named Leydig. So you also see these cells called Leydig cells or interstitial cells. These are the actual testosterone-producing cells. So upon puberty, they start producing testosterone, and these cells now only, I'm looking at the very top, these cells release the testosterone. That testosterone only has to diffuse a very short distance to turn these spermatogonia on, and then they start making sperm for the rest of their life. So sperm productions are in the billions per day by men. Okay, billions of, with a B, of sperm produced every day. Again, the sperm are going to enter into the tubule of the seminiferous tubules, tail first, flagella first, and you'll very clearly see that over there. Overall process, spermatogenesis. Now, from this space where I am now, this box, little round spherical cells, to creating what we recognize as a sperm is a rather dramatic change in shape and form. And that little bit, it's the last thing I'm going to show you for now, the process of going from a spherical cell to what we recognize as a sperm is spermiogenesis. It is a subset of spermatogenesis. Spermiogenesis is a subset of spermatogenesis. And there's 4 things that happen here that I want you to appreciate, maybe 5. Number 1, the nucleus, right? Remember, this is already haploid, only has 1 of each chromosome, gets shoved up to what becomes the head of the sperm. So the head has the little nucleus with the DNA in it. Around the nucleus, remember there's the Golgi and the endoplasmic reticulum. Well, the proteins that these cells are making are all involved with burrowing through the egg. And all of those enzymes that are being made get packaged up also right along the head of the DNA in a little bag of enzymes called the acrosome. The acrosome. Now, you know acro means extreme, right? Extremity. So a little body, zome, at the extreme end of the sperm. And we'll see that upon fertilization, those enzymes are released and they're necessary to burrow through the outer layers of the egg in order to fertilize. Thirdly, the mitochondria, which are shown here as big blue blobs, end up lining up only in what's called the midpiece, or the neck. Some books call it the neck of the sperm. And then finally there's a flagellum that sprouts out on the other end. okay, the flagellum or the tail. So you have a head, a neck, and a tail, or a head, a midpiece, and a flagellum. Any of those terms are fine, they're interchangeable. So those are all the things that happen at the very end of the formation of the sperm in spermiogenesis. Now, like I said before, once the sperm are released here into the lumen, they don't yet swim well. So they're kind of washed away, and they're being washed toward the semen, the epididymis. Okay, and then mature there, grow up there, learn how to swim, and then are either resorbed, that is broken down by the body, or are ejaculated. Okay, they don't just continue to build up. The numbers would be overwhelming very quickly. So those sperm are broken down. Any thoughts, questions, or concerns over male reproductive system? Okay, so you're going to turn to your page in your Amerman, page 660, and there were just a couple of things to cross off, as I recall. The cremaster muscle you're crossing off, I forget the number. We can't see the cremaster muscle on our models, and you were also crossing off, I think, the tunicas. Other than that, I think everything was on the list. So start looking at your model. There are only 2 of the hanging boy models in the room, so please, if you have the hanging boy model, take a look at that. I'll show you a couple of things that are on that model that aren't on the other. Make that your priority and pass it on to the tables who don't have that particular model. As I continue with the female reproductive system, make sure you have your female model out. And before I do that, though, let me remind you there's 3 slides over there, and one of them, I've already described what a couple of them look like, but one is the cross-section of the penis, and again what you're going to see is what it looks like 2 eyeballs and a wrinkly mouth with something around it. So again, the 2 eyeballs are the corpus cavernosa, the blood-filling regions. The little squiggly mouth is the urethra, surrounded by the corpus spongiosum. Okay, and again, the histology, the page is on the board, and just don't forget to look at that, and I'll go over what to expect also in the exam next week. Okay, so the female reproductive system, more complex, has a lot more going on than the male, although I think you'll have as easier time with it than the male as far as the structures go. So we have, of course, the production of the sex cells, the egg, but also far greater purposes in the transport as well as the development of the offspring. In the female, we have the ovaries. The ovaries would be the female gonads. So we have the testis or the testes in the male, the ovaries in the female, and they would be, from an anatomical standpoint, considered the primary sex organs. The vagina and the penis would be the primary copulatory organs, but as far as making of sperm and hormones and all, that's the job of the gonads. So everything else is really just fancy plumbing. So the accessory organs, the internal and external structures that allow for that egg and to be released, or for the sperm to be released, that's all just fancy plumbing. So this image looks a lot like your model in front of you. If you take the uterus off your model, it looks just like this. Let me just point out those structures which have caused a little bit of trouble in the past. So we'll start anteriorly. So externally, all that can be seen externally in a female is called the vulva. That's not labeled here, but you'll see it, V-U-L-V-A, the vulva, and that's just what can be externally seen in the female. It's going to include the mons pubis, which is this region over the pubic symphysis with the majority of the pubic hair, and then 2 folds of tissue, the labia major and the labia minor. Now you'll see minora and majora, you can put that, but minor and major is fine. And then you see labia plural or labium singular. So any of those combinations is fine. Labium majora, labia major. And now things that are not as visible, the clitoris, the erectile tissues of the female, then the urethra going up into the bladder. Remember that this band of muscle here would be that urogenital diaphragm, and on either side of the urethra would be the external sphincter, the internal sphincter being up at the base of the bladder. Then the vagina, going up to the narrow neck-like opening to the uterus called the cervix, and then into the uterus. Now it's difficult to appreciate at this sagittal cut, but there would be 2 ovaries, one in front of you coming out of the board, one behind in the board, with the fallopian tubes and the 2 ovaries. So here we have the fallopian tube. It's also called the uterine tube or the oviduct. I'm okay with all of those. At the end of the uterine tube are a series of little finger-like projections called the fimbriae. And the fimbriae are like little vacuum cleaners. They're going to suck up the egg upon ovulation and draw it into the uterine tube. In the very background, I can see the ureter coming down. See a little, right in the background, little ureter coming down, and it's going to eventually go down to the bladder. And what would this be? This tube is what? Which intestine? Think about the section that we're in right now. So here's anus, right? Anus, rectum, sigmoid colon. This would be the descending. So that is part of the transverse, right? Going in and out of the board, if you will, right? Left to right. And that's about it. Now, there are 3 layers to the uterus. We'll talk about them a little bit more in lecture. The inside layer that sloughs off monthly after puberty is the endometrium within. And you know that METR as a root means uterus, the uterus or the womb, right? So we have the endometrium, the inside layer. The muscle layer is the myometrium. This all makes sense. That gets much thicker, much more muscular during pregnancy. And then the outer layer going all the way around the uterus would be called the perimetrium. So the only difference between a male and a female abdominal pelvic cavity really is the presence of this uterus, right? The male looks largely the same. I mean, you have a lot more external structures out here, but still an anus, still the urethra. So what's different here is just the uterus. So the uterus is going to be held in place by a large ligament called the broad ligament. Okay, so it's kind of holding the ovaries and the uterus into their proper anatomic position within the abdominal pelvic cavity. So here, uterus, the narrow neck of the cervix, the muscular uterus, and then coming off on either side, a uterine tube. And that's a hollow tube. And it's going over, and it does this little loop-de-loop, up and over, and then little fimbriae at the ends of the uterine tube. The egg will release the ovary, turn that around. The ovary will release the egg upon ovulation, and that egg will get scooped up into the uterine tube. It is possible, and probably not that unlikely, that eggs do not get scooped up and end up out here in the abdominal pelvic cavity. So there could be eggs here, right, in the abdominal pelvic cavity. And sperm also could come up through the flute tube and also come out in here and actually fertilize that egg. But that embryo would have no chance. There's no place for it to adhere, no place for it to develop. So we know that fertilization happens far more commonly than pregnancy. Fertilization is simply egg and sperm coming together, embryo is formed. Pregnancy is when that embryo then adheres into the uterine wall and sets up a 9-month stay. That's a big difference. So fertilization can happen out there in the abdominal pelvic cavity. Fertilization can also happen in the fallopian tube and then get stuck in the tube. That'd be called an ectopic pregnancy, where it gets stuck in the uterine tube and can't progress to the uterus to grab on. And that can be a very serious situation where the tube can rupture if that embryo does get large enough and causes some damage. Okay, I had a question earlier about the timing of all this. So fertilization, sperm are coming up, right? And sperm are good for about 72 hours. So you got a 72-hour window for the sperm, but the egg being released is only good for about 24 hours. So that means that sperm had to have been present 2 or 3 days before ovulation, or get there quickly after ovulation, and you have that 3 or 4 day window of fertilization. That's possible. Then the embryo is formed here in the fallopian tube, and it's about a 5 or 6 day journey for that embryo to come down and adhere onto the wall of the uterus, onto the endometrium. If we cut into the ovary, as is done over here, we'll get something like looks like this. Okay, and it's very simplified, and you'll be looking at the ovary under the microscope today. Now, there's also a ligament, a ovarian ligament, that connects the ovary to the uterus. That is a structural type connective tissue. I told you back in bone world that ligaments connect bone to bone. And that's true. But ligaments, the definition expands a little bit. Ligaments can also connect structure to structure. So the broad ligament and the ovarian ligament are holding structures in their proper place. And the ovarian ligament specifically is holding the ovary over to the uterus. So how would you describe the location of the ovaries based upon this image? The ovaries are what? Lateral and toward the more superior border of the uterus. And structurally, what do we have in ovaries? Just like the kidney and just like the brain. The outer layer would be called the cortex, and the inside would be called the medulla. We've seen this over and over. So it's the same idea. Okay? And functionally, what do ovaries do? They produce eggs, right? Now those eggs can be released from the body. So that makes the ovary exocrine. Things are being released from the body. But the ovaries also are endocrine. They release hormones. And so that means that the ovary is mixed, exo and endocrine. And that's true of the testes as well, right? Sperm being produced for outside the body and testosterone for the bloodstream. What other organ have we talked about that is both exo and endocrine? Along the way, digestive. Releases hormones, but also releases stuff out of the body. Pancreas, right? The pancreas is making insulin, hormone that we've all heard of. And also, though, the pancreas is making digestive enzymes, which are released into the duodenum and therefore out of the body. Okay, so we've got the pancreas also as a mixed endo-exocrine gland. Okay, just a couple features about oogenesis. So we had spermatogenesis, that was in the male, the production of sperm. In the female, we have or oogenesis, the production of eggs. Very different in the number, though. This does not begin until puberty. As far as the release, about one egg per month and then the ovaries are kind of taking turns. Now, it's not left, right, left, right. It's not a perfect left-right alternation. But basically, every month, one of the ovaries is a little bit further ahead of the other one, and it sort of takes charge and is the one that month that will release the egg. And if a woman has lost one ovary, then the other ovary will do this every month. Okay, so it's not, if you lose an ovary, then the other one will just take over and do it every month. If you have both ovaries, they're kind of taking turns and kind of being polite about it. It's more than that, but we can just think of it that way for now. Now, the difference here is that the eggs are already produced in the female's body long before she's born. That's very different. In the guy, the sperm are not being produced until puberty, until testosterone is being produced. No sperm at all prepubescent. In the female, the eggs are already there. By the 5th month of fetal development, the eggs are already there. They've already been made. The eggs were produced by cells called oogonia. Well, that sounds a lot like the spermatogonia cells that I talked about that were in the seminiferous tubules. But they already start their maturation process. They all get used up, right? So there's a limited number of eggs that a female will have in her body that can be produced, whereas the guy, those spermatogonia are being regenerated constantly, so sperm production stays in the billions per day. Now, these eggs are very important. And just like something that's precious needs to be protected. So around these little eggs is a bubble of tissue, other cells. And that bubble of cells and tissues collectively is called a follicle. And these eggs are going to be protected in this bubble of tissue for the next 20, 30, 40, 50 years. Right? They have to produce long before you're born, ladies, and then are saved and protected until they wake up later in your life. Now, these primordial follicles, these little eggs with follicles and bubble wrap, are going to be found in the cortex of the ovary. So around the outside edge is where you will see these little primordial follicles packing into the ovary. Now, here's a simplified picture of this. So here's just a little bit of the cortex between these two lines. And here I'm seeing 6 or so of these primordial follicles. This is way oversimplified, but it gives us an idea of what's going on. Now, once puberty begins and hormones are being produced, then on a cyclic way, every month or every other month, about 20 or so of these primordial follicles are going to kind of wake up, and they're going to start a developmental journey as a primary and a secondary follicle. Now, as they move, they get bigger. The egg gets bigger, the whole follicle gets bigger, and basically it's a race. And only one of these 20 or so will win the race. The other ones will kind of just fall off, right? And they just kind of die off. One will continue to develop all the way and get very, very large. So that's all a huge follicle. The egg on the inside's gotten much, much bigger. It's surrounded by more fluid and another layer of bubble wrap around it, more bubble cells around it, follicular cells. That one that makes it to maturity will be ruptured out of the ovary in ovulation. Now you see that the egg is released and there are little layers of cells. That bubble wrap is still around it. And that's why when the sperm comes in, it has to release the enzymes that were in the head. And those enzymes are now going to break through those layers of the egg and allow for fertilization. Now, we're not done yet, because some of those bubble wrap cells are going to go on to become another very important structure called the corpus luteum. Corpus, body, lutea, yellow, the yellow body. And this corpus luteum is shown in two different stages here, and it's producing hormones, and we'll see why those hormones are being produced in a moment. After about 10 or 13 days, those hormones stop being produced, and now the corpus luteum becomes the corpus albicans. Think albino, white body. Right, so we have the yellow body and the white body. If I slice into a 7-year-old ovary, and a 7-year-old girl, I'm only going to see primordial follicles, right? No evidence of any other development going on. If I slice into a mid-aged ovary, I'm going to see primordial follicles, but I'm also going to see evidence of eggs in different stages of development. And the older it is, the more of these corpus albicans that would be present. They're like little scar evidence that a cycle has gone on, right? So an older ovary would have more of these albicans, and a younger ovary would have fewer of them, and a prepubescent ovary would have none of them. This is the ovarian ligament over here, so I know that the uterus is over here, right? So that's what connects the uterus to the ovary. I'm not going to go into all the details here. We'll save some of that for lecture. But the slide that you're going to look at, the closest one to me over there, you're looking at the outside edge of the ovary. So here's the outside edge, and the outside edge, this would all be cortex. And what you see is that packed in here are those little eggs with their bubble wrap. So those are all the little primordial follicles around the cortical edge, around the cortex. Keep in mind, this is going around the entire ovary, not just in the little section that you're seeing. Now, I know that this particular ovary is taken from somebody post-puberty because I see that some of them have woken up and are on some developmental journey toward becoming mature. That's all I can really tell here, right? Now, eventually, this is all under hormonal control, and we'll deal with those hormones in lecture next week, so I'm not going to talk about that right now. Eventually, this is the big follicle that's going to get released. And you see how big it gets. I mean, it really gets large. So there's the egg inside. There's a little nucleus of the egg in the very, very center. And this is the fluid and the other bubble wrap that goes around it. And that egg will be released upon ovulation. Now, just like I told you before, there's going to be a couple of details I want to share with you now just to get you thinking about the reproductive lecture that we're going to have. When we get there next Thursday, structurally, it won't be a problem. Again, it'll be the same structures we talked about here on the models. What will be different is a few numbers and a few timing issues. And one of the things that I'll finish up with next Thursday will be a discussion about ovulation and about menstrual cycle. It'll be a very introductory presentation, but I will introduce some of the hormones and sort of the big picture of what's going on. So let me just 2 minutes on this, and then we'll be done, and we'll look at the female models. Across the top, this is what's happening in the ovary post-puberty. So I start off with a bunch of these primordial follicles. They're sitting around the outer edge. We don't understand why, but about 20 or so of those every other month will be awoken and will start a developmental journey. They get larger, And of the ones that start this process, only one will make it to the finish line. The other ones will fall off along the way. The one that makes it to the finish line will be released from the ovary at a process called ovulation. Now that's going to happen right about the middle of this 28-day cycle. So we've got a 28-day cycle, and day 14 is ovulation, right in the middle. Now some women have shorter, longer, irregular periods, but we're going to make 28 days the normal cycle. Now after that, the bubble wrap around the released egg becomes the corpus luteum. It'll stay for about 10 days or so, and then eventually will regress to the corpus albicans. So that's what's going on in the ovary every other month or so. What's going on in the lower panel is what's happening in the uterus, the uterine cycle. These are connected through hormones. And 28-day cycle, the first 5 days menstrual flow. The downward, what I'm drawing is that the actual thickness of the endometrium is sloughing off. So the inner layer of the uterus is sloughing off. That's the endometrial layer, part of it. And there's bleeding involved with that. And then for the next few days, we're building the uterus back up to its premenstrual layer. This is called the proliferative phase. Think about the Russians, right? They're proliferating their nuclear arms right now against the Ukrainians, right? So they're building up their arms. So we got the proliferative phase, building it back up. And then at the end, we have the secretory phase. And that is when you go out for Thanksgiving, you want to have a little study binder with all the major slides, all the major images from your Amerman book. So this page reminds us, this review week number 2 reminds us of what we've been doing. So week 6, that's where the second lab exam will start. That was back at the nervous system. We did the eye, the ear, the brain, the spinal cord, and we named a bunch of nerves on flat Stanley. So those are the models you need to be familiar with. And then we also looked at 2 things under the microscope that day. You saw the neuron, right? Parts of the neuron model as well. And there was also the cross-section of the spinal cord. Remember that image? Pretty clear image of the spinal cord. Then the next week was the digestive system. We had the big digestive model. That was also the day that we introduced the location of the endocrine models. So we saw the testes, the ovaries, the pancreas, the thyroid. Much of that has become second nature since we introduced it back in Lab 7. The only histology that day was the cross-section of the small intestine. Remember the layers? Mucosa, submucosa, muscularis, serosa. Lab 8 was the beginning of cardiovascular. That was the blood and the blood typing and the heart. The flow of blood through the heart. And that was also, there was a microscope set up, I don't think anyone looked at it, of cardiac muscle. Remember, cardiac muscle has the intercalated discs. I think we've got our eye trained for that. If you saw it again, you would know what it was. The next week was the second part of cardiovascular. It was the vessels, and we also introduced the lymphatic organs that day. So make sure you know your arteries and veins and know your lymphatic organs. There was no histology that day. Then the next week, which was just last week, we had the urinary and the respiratory systems, and you're fresh with those. Last week there was a little bit of histology out. I don't think anyone looked at it. It was up on the board. There was a slice of the trachea. Now, if you're looking at a slice of trachea, what are you going to see? A big airway, right? The big airway with cartilage around it, hyaline cartilage around it. And recall that that airway would be lined by pseudostratified columnar with cilia. And then right behind it would be the esophagus, right, with stratified squamous, non-keratinized. Also over there were a slice of lung, and you're looking at little alveoli, little air sacs, and recall that's going to be simple squamous. And then here we are today, and we've got the reproductive systems, and there are 4 slides over there to look at histologically. There was the seminiferous tubules of the testis. There's a slide of sperm. There's a slide of the fetus, the fetal penis, and then finally there is over there a slice of ovary looking much like what I just showed you with the outside, the cortex having all the little primordial follicles, and then you see some larger follicles developing within the ovary. Okay, so that list is not completely exhaustive, but it's a good place to start. Now, also to help you with this, if you haven't noticed, but if you go on to Blackboard and go under our course, let me pull this up for you. There are two. Okay, so make sure you take your PAL quiz. Okay, that is everything I need to squawk at you right now about. So let's look at female. It won't take very long at all. And I'm going to start getting ready for the dissections here in a moment.